Welcome everybody to another Beyond Terrain mini lecture. Gorgeous day today. Great day for a little walk and talk. We're going to be discussing misconceptions in microbiology today. This has been kind of a long time coming. A lot of thought and research going into this topic. Not something I see too many people discussing. Something I've discussed with a few acquaintances online. But very important topic nevertheless. And what it's going to do is going to set us up really well to discuss pleomorphism, and we will discuss pleomorphism, the changing nature of microbes, a little bit in today's episode. But before we get started, why don't you subscribe and like? Great way to help support the channel. And if you really want to support the channel, you should consider joining the Beyond Terrain community, a place where like-minded individuals come together, where we can move past these broken systems. We are currently in a waitlist right now. You're not going to want to miss the launch. Founding members are going to get a ton of benefits, locked in low price, free complimentary consultation to help shape the community, discuss goals and your terrain strategy for implementing it into your life. And that's really what this community is about, implementation, right? We know these things, and we're going to dive deeper into these topics, more specific topics. But we also wanted to work on implementing these things. You know, you can know as much as you want about terrain and alternative health and blah, blah, blah. If you're not living the life, if you're not putting these things into action, you're just not going to see results. And we've all been there, and this is really the hardest part. So that's what the community is focused on. So make sure you go and check that out. I'll leave the links down below. Join the waitlist. Today we're going to be discussing some problems in microbiology, especially in regards to microbial species. This is kind of an area that you can get hung up on with pleomorphism. I had so many questions regarding this. I'm like, which species responds to which environment, et cetera, et cetera. And it can be really complex, and it's not that it's an unanswerable question, but the way that we have to go about this might be a little bit different than what mainstream microbiology teaches us. You know, originally microbiology is built on classification of, traits, observable things under the microscope, their observable anatomy. Now microbial species are totally based on genomic methods. And to identify them, we use serology, immunology, we use antibodies, right? Which we know are the cause of the reproducibility crisis in biomedical research. They're just totally unreliable. You know, a lot of modern microbiology now relies on cell cultures, totally unnatural environments. And so of course, in discussing this, you can hear a lot of the overlap with virology, which we know the methods of virology are circular. We know that they're absolute nonsense. We know that they're totally unscientific the way that we go about it. And the overlap here is really important. That's what kind of stemmed this little exploration into microbial species. Obviously, there are certain things that we do know about microbes that, are kind of irrefutable. So we're going to cover that real quick. We're going to kind of unpack a lot of the problems in modern microbiology. We're going to talk about a few of these key laboratory conditions or interventions that are problematic in deciphering what we know about microbes, what we know about their species, etc. And then we're going to kind of end with a little bit of a reframe. We're going to kind of bring things together and try to come up with some facts about microbes that we can work off of. And I'm going to try and remain empirical here. Obviously, we need to start with good foundations. We need to start with valid premises to be able to work off of these in a logical, rational format. So what are the foundational things that we know about microbes? We know that we can see microbes in a live state. Now, in electron microscopy, this is not live state, right? So we know that through Harold Hillman's work, a lot of what's done in electron microscopy is just artifacts. It doesn't represent real life whatsoever. There's so many added steps, which again, this overlaps with virology. We have to stain, dehydrate, freeze, rehydrate with a chemical compound. There are so many variables going on. Even in the use of a lot of light microscopes, they still use dead, stained tissue samples or microbial samples, right? When we're talking about microbiology, when you see the picture of a pink slide, when it's pink, it's stained, right? So you're looking at it in a dead state. When it's in the live state, you're either using a dark field microscope, which is kind of like that black background, or a white background, and everything's really faint, right? It's almost difficult to distinguish certain things. Obviously you can see some of the superstructures like flagella, or whether they're a rod, a circle, different observable shapes of the microbes. When we're talking about things that cannot be refuted in microbiology, it's things that we can observe directly through microscopy of microbes in their live state. I'm not going to sit here and say that stained samples or electron microscopy is totally useless, but you know, there's so many assumptions that need to be made that it doesn't really fit into our discussion right now. You know, it's full of artifacts, it's full of uncontrolled variables, so we're going to leave that totally out of our discussion. And of course, when it comes to microbiology, especially in the earlier years, how we classified microbes was totally based on what you could see under the microscope, right? So when you called a species like a streptococcus, you were seeing a distinct structure under the microscope, and that's what we called that group, right? So if you look at the history of microbiology, we didn't have these millions of different names for microbes and subspecies, and that's totally due to genomics, which we're going to discuss later. But initially you had sort of this quite refined class of microbes, right? But the problem was with appearance. This didn't tell us anything about function. It didn't tell us anything about what the microbes do in the environment, etc. Right? So you were only classifying these microbes based on their structure, right? Based on their anatomy that you could see under the microscope. So another thing that we know about microbes is that they're ubiquitous. We know that they exist absolutely everywhere in our environment. Obviously, we've created sterile environments. Like, we try to keep our hospitals and our homes and our schools as sterile as possible. So we know microbes are everywhere because of straight up microscopy. You take a sample of anything around in the natural world, you're going to find microbes there. Now what we use are, metagenomic sequencing. You take a sample, you run it through a machine, it churns out millions of little reads of a genetic sequence, and then it churns out about thousands and thousands of different microbial species that are in this sample somewhere, right? This is totally in silico. It happens all in the computer. Again, we can get into the circularity of this, how the conformation of these genetic sequences are confirmed by the processes used to determine the genetic sequences themselves, which ends up being one big circle. Okay, and the last thing that we absolutely know is true about microbes is their importance. They're absolutely vital to life and living systems. If you were in this forest right now and there were no microbes, there would be no forest. We know that microbes are vital for every single process that happens in human body, in animal bodies, in plants, in nature, right? Microbes are absolutely vital from the growth stage to the decomposition stage. Microbes and the micro life of our realm plays an absolutely vital role in all this. All right, let's go over some problematic foundational assumptions in modern microbiology. So the first major assumption is that microbes are fixed species, right? They're monomorphic. They have one shape and size, although some modern microbiology refutes this because it's sort of irrefutable when you look at microbes in their live state and you don't keep the conditions fixed. You could see them changing, even Helicobacter pylori, they say is pleomorphic, right? It has more than one shape. And they're starting to admit this with more and more microbes because it's kind of undeniable, right? The reality of the situation is that there are no fixed microbial species, right? Species are a result of the lab. What we see in the lab are fixed conditions. We see sterile conditions. We don't see the diversity or the complexity of our realm of nature. So microbes are actually pleomorphic. They can have multiple shapes and they can actually change from species to species depending on the environment. There's actually a body of literature on the fluidity of microbial species. Sonnen Sorea's book, actually, A New Bacteriology, really set the foundation for this. And he was, a pretty modern type of guy. Even in his book, he talks a lot about genetics and how microbes should be viewed as just a genetic pool rather than fixed species, right? He was saying that microbes can change just like that. And a lot of the modern assumptions like horizontal gene transfer, all of these different processes that microbes have actually kind of support his theory, if we're going to agree with the whole genetics thing. Genetic variability in microbes is huge. I mean, E. coli strains can vary by 30% of their genomic sequence. Obviously, these numbers are all based on so many assumptions, but let's pretend they're true. What we're seeing is a total mismatch of genetic sequences in microbes, right? Nothing is fixed, and that's what we see when we talk about variants too. You know, we have all these variants and subspecies. Well, this is due to the microbes actually just changing species in regards to the environment, right? They're just constantly adapting to their environment. So a lot of the modern theories that we have, which sometimes could be interpreted as backfill, as ad hoc reasoning, are actually supportive of this whole idea that, you know, microbes are not fixed species. So another pretty obvious assumption is that, you know, there's one microbe that causes this specific disease. We know that the germ theory is nonsense. We know that our empirical observations, our controlled contagion studies, all go against this idea. They prove a direct falsification of germ theory, especially the one microbe, one disease idea, right? This is total nonsense. Even a lot of in vitro studies go against this, which is something that we'll be getting into in the future as well. And obviously, reproducibility is an issue, right? So even in the laboratory, what we find is that there are many problems with reproducibility in microbiology. That's why we need to keep everything so stable, so sterile. So a lot of what we find in the laboratories is totally opposite to nature, right? The only constant in nature is change. Things are constantly changing. Things are constantly moving. I mean, look at the water behind me. It's just flowing and flowing and flowing. These trees are growing. The moss is changing. You know, everything is constantly changing. The environments are constantly changing, and that doesn't happen in a laboratory. The laboratory cell cultures are also kept sterile, which is something we'll discuss in a little bit here too. But the fundamental idea, you know, is that laboratory conditions equals reality, which is, again, how do you prove that? How has modern microbiology proved this? How has modern science proved this in any respect? You know, what happens in the laboratory often opposes what happens in nature. Like, the laboratory conditions are totally against nature. They're totally opposite to nature. So a lot of what we find in laboratories are actually just artifacts of the laboratory. Same goes for microbiology, right? Fixed species, monomorphism. We look at dead microbes. We look at dead tissues. We look at stained tissues, dehydrated tissues, frozen tissues. When you look at things in a live state, you get a whole different picture of what's actually going on in nature, right? Why would we study things in their dead, decrepit, dying state? We need to study things in their live state. You know, the assumption, it goes back to Hippocrates. You know, even they were asking questions about, why would we cut open a dead body? What does that tell us about a natural, living human being, right? So obviously there were moral reasons for that. And Dr. Berlando gave a great point on this. You know, it can tell us something about anatomy, but no more, right? All you can know is anatomy from this. And with anatomy, you still need to be careful of artifacts like electron microscopy. Introduces so many artifacts. Think about cell culture. Harold Hillman, once again, make sure you go look into that. We'll be talking about that more in the future as well. One of the foundational techniques in microbiology is obviously the cell culture. Now, the cell culture obviously has many problems. It lacks whole body complexity, right? We're not even close to what's natural, right? Like we were just discussing. This suppresses natural behavior. The opposite conditions of nature is going to give opposite answers. You know, so the opposite of a pleomorphic microbe is a monomorphic one. And that's the reality of what we get in the laboratory, because it opposes nature. You know, in the cell culture, what we have is fixed conditions. We have perfectly controlled pH with pH stabilizers, temperature, sterility, right? We're introducing antimycotics, antibiotics. Microbes and fungal cells live hand in hand, even with algae cells, even with tissues, plant or human, right? All of these things live together. That's why you need human tissue or some sort of animal tissue or plant tissue to be able to grow microbes, because it's a necessary component, right? So still what we're doing is keeping it at very stable conditions, which is totally unnatural. Stable and sterile, that is the crux of cell culture. And we could dive way deeper into this topic, but I think we'll leave it at that for now since we already kind of discussed it. Another thing with cell culture, it's like crazy because we can only culture 1% of the microbes that we know about. That means 99% of the microbes in our realm cannot be cultured. Now, obviously, these numbers are based off modern microbiology understandings, but still, let's assume that everything we know about microbiology is true. We only know about 1% of the microbes. What does that tell you about what you can know about the other 99%? We can't culture them. We can't study them in a lab environment. And the reason we use laboratory environments is because you cannot study these in nature, in vivo. You know, you can't set up a microscope of the gut lining in a live person or animal, you know? So what we do know about microbes comes from cell cultures, and only 1% of these microbes can be cultured. Let that sink in for a moment. The workaround, obviously, is metagenomics. And I think this is kind of what fuels that whole 1% and 99% number, because, again, it's like, if we're talking about species not necessarily being a fixed thing, what we're talking about is just nonsense when we talk about microbial species. 1%, 99%, it's something to consider in the modern narrative, but it's mostly based on metagenomic input, right? So we take environmental DNA, we put it in the computer, it generates all these little reads of genetic sequences, and all of a sudden you have thousands and thousands of different microbes in your sample, all based on probability, all based on our computer sequences, all based on human assumptions. And it doesn't even matter that the computer knows this too, because the computer only knows what humans know at an accelerated rate. The computer is based on all the same assumptions that humans make, right? Because computers run off human input. Now, what we try to infer from these genetic sequences is function. We try to infer traits. We try to infer as much as we possibly can. But the reality is that none of this can actually be confirmed, especially if we're talking about unculturable microbes. You know, so how are you going to test this genetic sequence that you found in this random sample of water in a lab if the microbe is unculturable? How do you verify it even in artificial laboratory conditions? Well, you can't. So again, you can see how this is just falling apart more and more and more. Our genomic data is totally based on interpretation. It's not based on observation. These are indirect interpretations of what's going on in the world. And it's based on a whole other plethora of assumptions in which we're going to discuss more in the future. These are big, big topics, especially genetics, but breaking that down, a lot of people are doing it now, so make sure you stay tuned. Obviously, we know that Koch's postulates is total nonsense again. Koch's postulates has never been attained for any microbe to ever be named, to exist, whatever. If it's found at the site of disease, that doesn't mean it's causing disease. That's correlation. The reality is that it's cleaning up disease. Here's another good question. If you can't culture the microbe, how could you even do a Koch's postulate study in the first place? Big old question that remains unanswered, yet we still attribute cause to disease to microbes that are unculturable. Total nonsense. Even culturable microbes have never actually been used in a Koch's postulate study and achieved positive results to support the germ theory. Additionally to the unculturable microbe problem, we also have this other issue that comes in laboratory that can be combated but adds this layer of difficulty and layer of assumptions to the whole basis of microbiology. This is what we call a viable but non-culturable state. Microbes can enter this VNBC state where they're technically alive, but we can't grow them in a lab. Now, they say that this is caused by stress, by toxins, starvations, etc. That's why we need to keep the laboratory conditions super tight, super stable, or else they might enter this state of non-culturability. Now, pretty well any microbe can enter this state. That's why we have to keep these conditions so stable. Otherwise, things are going to start to decay. We artificially keep this stuff alive, right? The other interesting thing about this state is that they say that these microbes are still metabolically active, but there's so many problems that come with this state, like PCR, reverse transcriptase PCR, stains, antibodies. All of these things become totally unreliable here. When something's in this VBNC state, what actually happens is these techniques stop working as they should, right? PCR becomes totally unreliable. what happens is these microbes are kind of receding into themselves, kind of going, you know, I don't like these conditions. We got to hunker down and just kind of do what we got to do to stay active and alive. So PCR stops working. Antibodies becomes less reliable than it already is. Again, this kind of contributes to this whole idea that presence doesn't mean pathogenicity, right? It doesn't mean that the presence of a microbe is causing illness. If microbes in a lab can enter the state, you know, modern microbiology should assume that this happens in the body. Now, I'm not sure if it does happen in the body, but again, it's like, how do we even go about determining whether or not this happens in vivo or not? If you can't test with PCR, if you can't test with antibodies, if you can't use a microscope to, you know, verify the presence, how are we determining whether or not this is just a laboratory artifact or not? This is obviously a huge problem that happens in microbiology, and really kind of shits on genomics as well. Because PCR, again, fundamental. It's absolutely fundamental to modern microbiology. All right, so what about microbial genomes and evolution and all these genomic techniques? Again, it's just like problem after problem. Really, microbiology, modern microbiology, especially the classification of all these different species and subspecies, all relies on genomics. A contradictory observation is that there's major variability within species. I mentioned E. coli earlier, 30% difference in their genome than another species of E. coli, right? We have these major variants within the same species. This totally contradicts microbial genomics. You know, there's obviously a lot of contradictions in genomics, even in humans and animals, but for microbes, 30%. They say chimps are 2% similar to humans, and look how different we are. Again, not saying that I agree with that, but that's what they say. A big topic could be horizontal gene transfer. We could dive so deep into this. Part of me wants to say that it's just speculative backfill to kind of fill up this whole gap in the genomic narrative around microbes. How do we prove horizontal gene transfer? How do we prove gene transfer between microbes at all? You know, these are great questions. Now, there are so many methods of gene transfer between microbes, different types of reproduction, how they go about reproducing. Some divide, some are asexual, some are sexual. It's like a very complex topic. I don't know how most of this stuff is proven, especially based on all of these assumptions and problems in the study of microbiology already. You know, the reality is that a lot of this stuff is just going to be laboratory artifacts. And if we're saying that all of this is based on the genetic variability, that's based on the idea that our genetic methods can actually determine the sequences of these microbes reliably. You know, and there are people who realize this, like Soren Sonea. He has a great book, and it's all about how there are no species in prokaryotes. You know, he says that microbes are just a global gene pool. They all just share genomes. They all just change and adapt to the environment as you go. So you don't even have to reject genomics to accept this either. I don't really care what you think about this, about genomics, whatever. Regardless of whether you accept it or not, it still doesn't support this idea of monomorphic, specific species. You know, there is constant gene transfer going on, even in the modern understanding. You know, it's total adaptation to the terrain. All of these observations seem to support the terrain and seem to go against this monomorphic bullshit germ theory narrative. Like, all of this supports pleomorphism. All of it supports adaptation. So, and again, foundationally, we can talk about PCR all day and night. There's some great people that talk about PCR. Lots of great resources. Jamie Andrews has a ton on PCR. Cornelia Tomzik, we actually interviewed her. Ton on PCR. Very scientific evidence against the use of PCR as well. Podcast coming out very soon with her. So I won't do it justice here, but we know that if anything, PCR is good for just detection. There's no proof to PCR. You know, and again, we get into this circular reasoning of like, you PCR a stream of water to detect whether or not there's a pathogen in it. How do you confirm it? Well, you confirm it through microscopy. Well, microscopy actually can't confirm nor deny the type of species because all you can see is structure. So you use antibodies and serological assays to confirm whether or not this species is there or not, but antibodies are unreliable, and how do we determine which antibodies are for which species? And so we use PCR again, and again it's this big circle. So we know microbes exist, but like it's hard to just it's hard not to say we know microbes exist, but we don't know jack shit about them. At the end of the day, genomics, especially with microbes, but realistically you can kind of extrapolate this to the entirety of genomics. It's just circular. It's circular, especially when it comes to function. Nothing with genomics can reliably tell you about function or anatomy, because all of it needs confirmation through the processes that form the premises, right? So this is the definition of circular reasoning. You know, if your premise is microscopy, but you can't know anything using microscopy, you need PCR to confirm, and you need, you need serology to confirm. But to confirm the findings of serology and PCR, you need microscopy. Again, it's the same circle as virology, just for microbiology. So what do we know about microbes? Well, we know that they exist. We know that they're absolutely necessary for life. Sterile environments always cause disease. You take the microbes out of the forest, out of the human body, and it causes disease. We know that antibiotics wipe out microbiomes, and this comes with so many problems. Low microbiome is associated with every single disease that we know of. Whether it's an acute disease, an acute infection, whether it's a chronic, long-lasting degenerative disorder, whether it's a psychological thing, microbiome has to do with everything. You know, so how are we measuring microbiome? That's a whole different question, whole different story that we're gonna have to get to in another video. But we know that microbes are important, and we know that we can see them. We can use live state microscopy to see microbes. I think that we can reliably say that all microbes are pleomorphic. I say this because all it takes is a dark field microscope. You can see that microbes are changing form. They're changing species even. You know, they can even change from a bacteria to a fungal cell. You know, we know that it can change from a small little tiny particle to a rod to a circle. You know, it could change form and shape. All this can be observed through dark field microscopy, and there's observable evidence of this. You can actually see this. There are videos online of dark field where you can see the changing nature of microbes. I think without a doubt we can accept pleomorphism and the pleomorphic nature of microbes. And more, microbiology is starting to accept this because they have to. You cannot reject it. You can't reject the truth forever, right? The second a scientist looks under a dark field microscope, they're seeing the changing nature of microbes. You can't just reject that. I also think that we can know that microbes bioaccumulate toxins. We can test the quantities and concentrations of toxins in microbes versus host cells. A lot of this can be tested pretty close to in vivo, especially in the aquatic realm. There is a beneficial role to what we call pathogens. Again, a lot of this is done in fish. Like, we know that parasites in fish are extremely helpful in removing the heavy metals and kind of regulating heavy metals in the body. You know, we know that they have this bioremediation role. They take toxicity, they change it to a form that's more usable, that's more beneficial to nature, to humans, to animals, to plants, right? And I think that we can reliably say that microbes adapt to their environment. Microbes adapt to the terrain. You cannot grow an orange tree in Canada because it's not the right environment. A pathogen, right, a listeria microbe can't grow in a healthy environment. It needs to have the proper environmental conditions to be present, right? So a diseased environment precedes the bacteria, precedes the pathogen. Same goes for fungus, same goes for parasites. It's the same old story. I think that some sort of classification can be helpful for microbiology. I think it can be helpful to classify microbes based on traits, maybe even on function. You know, there is some validity to this. Even when you're looking at, you know, the old work on pleomorphism, on bacterial cyclogeny, on Gaston Naisson's work, right? They talked about different types of structures and species, and obviously they all have different names, whether you're studying Naissance or Enderlein or Antoine Bichon, everything has different names, but they were trying to name structures and classify them based on traits. Now, This is just classic concept formation. I don't see a problem with this. It's not harmful. I think it's helpful to understand that we're just talking here. You know, what we talk about doesn't necessarily encapsulate and encompass this beautiful dynamic complexity of nature. Whether it comes to discussing microbes, parasites, or even vitamins. Any part of science, right? It doesn't, when we're discussing it, it doesn't encapsulate this beautifully complex nature that is around us. Understanding this is really important. You know, it moves us away from this whole fear-based model. I think it moves us towards a greater understanding of science, especially microbiology and biology in general. A lot of these fundamental assumptions, you know, are present in cell biology. They're present in all of biology, really. There's a lot of overlap in biology. Virology, microbiology, cell biology, a lot of it you can extrapolate, right? So you can sit down, look at the methods that are used, and realize that there are a lot of same assumptions, a lot of the same laboratory conditions, interventions, processes that are just as harmful and kind of leading us astray as the rest of them, right? So a lot of work in virology done. Hopefully this demystifies a little bit of microbiology. I think we can kind of decipher some foundational principles as we did in this video to hopefully work off of and apply to our lives, right? And that's what it's all about, and that's what really led me to create this Beyond Terrain community. Application. How do we take what we know about microbiology, about true microbiology as discussed in this video, how do we apply this to our life? How do we apply these principles of adaptation to our life? How does that help us with health and healing and well-being? How does this translate across the fields? How does this apply to psychology, right? What are the underlying things that we know about this realm, right? There's so many things that we can know and so many things that are so confusing and just absolute nonsense and noise that we need to unlearn and ignore. And that's what the Beyond Terrain community is about. So make sure you go sign up for the wait list. If you're watching this video at a later date, go and sign up for the community. Come join us. It's going to be so packed with some great content, some great people. And we're going to be growing and become this beautiful, beautiful manifestation of the terrain paradigm. So I hope you guys enjoyed this video. Like, comment, let me know your thoughts. Do you agree? Do you disagree? I love to chat with you guys. And if you want to chat a little bit deeper in a little bit longer format, maybe even in a debate format to try and really pick apart these things that we're discussing here, join the community. I appreciate you guys for taking the time to listen today, so take care.